Welcome back, class. In today's lecture, we'll continue to look at integrated pest management. Let's start with a look at biological controls. Beneficial species are invertebrates that directly kill pest species. So beneficials are biological control agents that exist in agricultural systems. And obviously there are other beneficials such as those involved in breaking down organic matter so that nutrients are recycled. For example, earthworms, many types of beetles, mites and springtails. Beneficials can be categorised according to where they live. So are they resident species or transient species? They can be categorised according to the origin of the species, are they natives or exotic species, and according to the way that they feed, that is predator or parasite. So the relevant categories for us in IPM programs are resident beneficials, transient species, generalist predators, specialist parasites, and introduced or exotic species. The amount of living plant material in a crop or a pasture changes dramatically during a season, from almost bare ground to a dense canopy of plants. So a change from a herbicide-treated paddock about to be planted with wheat, canola or beans, for example, in autumn, to the crop just before harvest in spring is massive, and this is reflected in the number of invertebrates, both pests and beneficials, that can both exploit this same habitat. And the same thing applies to before and after grazing. So to the invertebrates, this represents a change in habitat structure and food sources. So different species will live there at different times. In summer and early autumn, very little green plant material is present. So this can't support the same number of invertebrates as in spring. However, there is a resident population living in the soil or on or near the surface of the soil, and this may include pests such as cockchafers, wireworms, European earwig, red-legged earth mite and lucerne flea. But it also includes beneficials such as carabid, beetles, predatory earwigs, spiders and predatory mites. And the number of these pests and beneficials are determined by factors such as the history of pesticide use, the current habitat structure and food availability. General predators feed on a range of prey, so populations of these predators can survive even when one type of food source is scarce. Specialist predators, on the other hand, their numbers will oscillate as the numbers of its prey either increase or decrease, as we can see here in this generic predator-prey cycle. So when the prey is a pest species, the aim is for us to maintain these oscillations of both prey and predator below levels where the pest can actually cause economic damage. That is, both are present, but at levels where neither are particularly obvious. That is, the system is in balance. Resident beneficials are the biological control agents that are relevant in controlling establishment pests in crops. Long-term maintenance of these beneficials is what is required to obtain sustainable control of establishment pests, and so disruption of these species in the previous crop should be avoided. Transient species are populations of important beneficials that do not permanently reside in the same paddock. These tend to be mobile species with relatively short generation times, that is weeks compared with months or years. And they tend to follow the movement of their prey or hosts. For example, these occur in a whole range of crops where different species of aphids form colonies. For example, parasitic wasps survive wherever their hosts occur. They'll colonise aphid colonies and then begin to increase in number. Another example, brown lacewings, and they're pictured here. They'll fly to wherever aphids are forming colonies and lay their eggs nearby. Another example, ladybirds. They respond to a higher number of aphids, while lacewings are found with even lower numbers of aphids. But they're no more or less important. They simply feed on different sets of pests. These transient species can be very vulnerable to pesticide use, depending on when the chemical is applied. For example, if there is one major flight of lacewings a year, lasting about 2 weeks following a flight, and an insecticide is applied that kills the lacewings in week 2, then no further large-scale replacement of that species will occur in the current season. Generalist predators are not restricted to one species of prey. So, for example, carabid beetles, like the one pictured here, are key beneficial species in pasture and cropping systems. They eat a wide range of pests, including slugs and caterpillars, and they also feed on dead invertebrates, which actually makes them vulnerable to secondary poisoning. That is, they're eating pesticide-affected invertebrates. Introduced or exotic pests and beneficials are those that are not native to Australia. Many beneficials have been introduced deliberately. Many pests have been introduced accidentally. For example, pests such as aphids, diamondback moth, slugs, red-legged earth mite, these all arrived from other countries as accidental introductions with plants or soil. So this means that they've escaped the natural enemies that evolved with them. Many native generalist predators will also attack exotic pests. Some specialist parasitoids of pests have also been deliberately introduced, and we can see an example pictured here. This shows Encarsia formosa, which was introduced to Australia in 2004 to help control the greenhouse whitefly. It's a parasitoid of late-stage pupa, and these whitefly pupa turn black when they've been parasitised. Let's take a look at some of our most important beneficials, starting with brown lacewings. Brown lacewings are native species. They're abundant in low-growing crops and native vegetation. They have delicate lacy brown wings, which you can see pictured here in the adult. They're 8 to 10 millimetres long. The juveniles, also pictured here, have long, thin bodies and prominent jaws. The cream-coloured single eggs are laid flat on the underside of leaves. Brown lacewings prefer to eat aphids, but they will eat just about any insect of an acceptable size. The larvae are the main predators, but adults will also eat insects, especially insect eggs. The larvae eat their prey using their curved hollow jaws and essentially eat out the body contents of their prey. Larvae can eat between 100 and 200 aphids each in their short lives. At temperatures of about 25 to 30 degrees, a generation will take about 3 weeks from egg to adult. The adults fly at night and move into crops soon after emergence. They may move into crops following aphid populations. Normally they're found in spring and autumn. Carabid or ground beetles. This group includes many predatory species. And you can see the common carabid beetle pictured here. They have these typical large jaws that are pointing to the front. The juveniles are also predatory, and they're likely to be important predators of pests such as cockchafers below the soil surface. However, detailed knowledge on key species of carabid beetle in agriculture is poor, but this is slowly changing. Research done in the UK suggests that refuges called beetle banks that foster carabid beetles can be used by farmers to help control pests in cereal crops, and this has been widely adopted here. Currently, research is underway to establish the impact of native carabid beetles on introduced slugs. So, although they're most likely to attack caterpillars, beetles and earwigs, the knowledge is still so poor that they're best regarded as beneficials without knowing exactly what they do eat. Carabid beetles have a 1 or 2 year life cycle, so the impact of broad spectrum insecticides applied to broad acre crops or pastures is likely to be far more disruptive to this group of beneficials than the others which have shorter lifespans, such as the lacewings. Damsel bugs kill their prey by stabbing them with their curved mouth parts and then sucking up the liquid contents. Damsel bugs are slender, pencil-shaped insects, and the immature nymphs look just like the adults. They prefer to eat caterpillars, but they also eat aphids and other pests. The female lays her eggs by inserting them into leaves or stems of plants, often in neat rows of 10 or more. Next we have the shield bugs, otherwise known as stink bugs. These are about 12 millimetres in length. They are predatory bugs with sucking mouth parts. They have large spikes behind the head, which forms its shoulders. Immature nymphs, such as that pictured here, have red marks on their backs. Eggs are usually laid in clusters of multiples of 14 and have they're metallic black with white spines around the rim of each egg. The photos shown here show the eggs, the nymph, and an adult feeding on a caterpillar. Next we have our predatory earwigs, including the common brown earwig. These are important generalist predators in Australian cropping and pasture systems. The males and females have different shaped forceps on the tip of the abdomen. The males are larger, but these are different to the main pest earwig, that is the European earwig. The main feature of the common brown earwig is the orange triangle behind its head. They eat a wide range of soft-bodied species, including red-legged earth mite and Heliothus. Pictured here on the left, we have our pest earwigs, the European earwigs, and to the right, we have our native earwigs. In Australia, we have many species of predatory mites, with the easiest to recognise being the snout mites. All these predator mites have a characteristic pointed head, and the Anista species are the more important ones in cropping and pasture systems. They're about 3 millimetres long with a red body and orange legs. Pictured here is the very fast-moving Anistis species, and it's attacking red-legged earth mites. We also have many species of parasitic wasps that attack aphids and caterpillars, including several species of Phidias wasps. The adults are 2 to 3 millimetres long, slender, winged, with a very constricted waist and thin abdomen. Pictured here are mummies. Mummies are the shells that were once aphids but have been transformed into cases containing the immature stages of the wasp. They're usually round and a bronze gold colour. If the adult has emerged, you can see a round hole in the mummy. If insecticides are not used, then almost every colony of aphid will have these parasites present. They're very efficient at finding and killing aphids. The female wasps sting the aphid and lay their eggs inside the aphid's body. The maggot-like immature stage develops inside the aphid, eventually killing it and turning it into a mummy. The maggot then pupates and then cuts its emergence hole in the mummy, and the winged adult wasp emerges to begin the next cycle. Phidias wasps are present in crops where insecticides are not used, and they often keep the aphid population below a detectable level. Problems with aphids may occur when the parasite is killed by the insecticide, but some of the aphids survive. Many other species of wasps that attack caterpillars can be large or very small, and different species will attack different stages of the host. So, for example, either eggs or young caterpillars or larger caterpillars. And there's also many flies that parasitise pests in exactly the same way. One of our most important beneficials is the ladybird beetle. There are 4 species commonly found in Victorian crops: the common spotted ladybird, transverse ladybird, two-spotted ladybird, and the recently introduced Hippodamia variegata. Their main prey is the aphid, but they'll also eat scale insects, mites, and thrips. They simply catch the aphid and chew it up. Pictured here in the middle of screen is the juvenile ladybird, which looks obviously very different to the adult. Hoverflies. The adults are very recognisable as they hover in one place in the air, often near flowers. They often have yellow markings on a black body. The adults themselves are not predators. They feed on nectar and pollen, but they lay their eggs near aphid colonies so that there is food for the juveniles that hatch out. Spiders. All spiders are predators and prey on a variety of invertebrates. Pictured here are some of the common beneficial spiders in Australian crops. Any control or suppression of pests by spiders is the result of the whole community of spider groups, but very little is actually known about the role of spiders in Australian agriculture. Now let's take a look at cultural controls. Cultural controls involve management activities that make the environment unfavourable to one or more pests. But these controls may not be to primarily control pests. So, for example, they might involve improving the habitat for the natural enemies or making the habitat worse for the pest. Let's look at a couple of examples here. For example, Pimental in 1993 from the USA, where changing rotation practices in corn was responsible for an increase in losses to pests from 3.5% to 12%, despite a 1,000-fold increase in pesticide application. Stinner and House in 1990 showed that changing from conventional tillage to stubble retained and minimal tillage increased problems with establishment pests from such as slugs and wireworms. It's useful to point out, though, that the use of some cultural controls are not always beneficial. So, for example, providing nectar sources for beneficials may also provide nutrients for pests. Another example, rotations with legumes can massively increase problems with white-fringed weevil. The photos shown here show the adult weevil on the left and the legless soil-dwelling larvae, which are about 12 millimetres long. They're white to grey with a brown head, and they're feeding on roots and kernels. Let's now look at some examples of cultural controls. Tillage is probably the oldest method of pest control, although this was not the primary aim of the practice. It results in a habitat that's less diverse in terms of many invertebrates, including some of the pests. And this is done mainly through the removal of suitable shelter and food, and also results in physical damage to pest species. In this photo shown here, you can see a paddock that's been cultivated to the left, and to the right we have no-till and stubble retention. Another example of a cultural control is the burning of crop stubble to reduce pest populations. This works in exactly the same way as tillage, in that it removes the shelter and food of the pests, but it doesn't kill pests such as slugs and earwigs, because these are usually protected in below ground sites when the burning is carried out. Burning is not promoted as a cultural control in IPM, usually, due to other undesirable effects on paddock health, and also it results in the removal of habitats for beneficials. Crop rotation is also a practice that's been used for centuries. It remains a key method for controlling pests and diseases of crops and pastures. The principle behind this is that specific pests of certain plants will continue to increase when provided with a suitable host. And so problems become worse as pests and diseases build up when the same type of plant species or closely related plant species are grown in the same ground in successive years. So this cycle, this pest and disease cycle, is broken with a non-host crop or pasture, or it's left fallow. Another extremely important control is variety selection, and this has been an important role of plant breeders for centuries. And just like plant breeders, genetic engineers now are attempting high-tech methods to exploit genetic diversity. So, for example, lucerne that is resistant to aphids and aphid-borne diseases has determined what varieties are grown in some locations. The time of planting is also an example of cultural control, and we've already talked about this in the previous lecture. Location can also affect pest pressure. So, for example, there may be a district risk of barley yellow dwarf virus carrying aphids. Another example involves a low-lying paddock with a high risk of slugs. And our last example here, newly planted clover surrounded by red-legged earth mite infected crops. And while such paddocks may have to be planted, the degree of risk should be recognised. One of the most important means of cultural control is weed. And this involves not just applying herbicides after planting, but getting on top of weed problems a year or two before the susceptible crops are planted. Control of brassica weeds will have a major positive impact. For example, if diamondback moth is a problem. Control of capeweed will have a major positive impact if red-legged earth mite is a problem. Pasture cockchafers. These prefer different environments. So the red-headed form prefers long grass or stubble, whereas the black-headed pasture cockchafer prefers to lay eggs in bare ground. So in other words, an overgrazed area. So the complete opposite controls by cultural means. Another example involves intercropping or trap crops. These are sometimes used to attract pests, and once in place, they can be treated with insecticides or managed within the trap crop. And the aim here is to provide pests with a suitable food or host plant that is not the main crop being grown. So, for example, cotton farmers may plant lucerne strips to attract green mirids. These are bugs that are very common in crops. Another means of cultural control involves the provision of alternative food sources for the predators. So, for example, provision of flowering grasses in citrus orchards and vineyards, which will increase the numbers of predatory mites. Provision of habitat, so for example, planting grass in spring is currently under investigation in order to encourage lacewings to that particular area. Tree planting is often raised as a possible cultural control method of increasing the numbers of beneficials. But the problem is that the beneficials in trees prefer tree habitats, not grassland type environments, while beneficial species that prefer cropping, pasture, grassland type environments tend not to inhabit trees. Beetle banks. These work in the UK, but there are limitations in Australia. It's actually preferable in Australia to turn the entire paddock into a suitable habitat for beneficial species, including predatory beetles. So there's no need for beetle banks as the entire paddock is being used as a beetle bank. Physical barriers can be used in small areas. So, for example, copper strips to repel slugs, mesh to prevent access by moths, and it's possible to use narrow strips of native grassland as a physical barrier because grasslands often have far lower pest numbers than the adjacent pasture or crop. Now let's look at some examples of integration of cultural controls. Example 1, pasture management for cockchafer. These are common pests of pastures and some crops in southeastern Australia. The larvae of each species are the pests, and the feeding preference of the adult determines pest pressure and damage. Now let's start with the black-headed cockchafer, which prefers to lay eggs in bare ground, and it lays more eggs if dung is present. So manage grazing in the spring and don't overgraze. If the black-headed cockchafer is already a problem, then it's too late for this approach. In this case, spraying with insecticide or ploughing the paddock to expose or physically damage the grubs would be recommended. The red-headed cockchafer, on the other hand, are almost the exact opposite of the black-headed cockchafer in terms of their feeding preferences. They fly in winter and early spring. They prefer to lay eggs in long grass or stubble. The grub never surfaces, so insecticides are useless. There is a fungal pathogen available called biogreen, but it's not a quick fix. Grazing management is key, and ploughing may be necessary in badly affected areas. Our next example involves cultural controls in canola, and there are several controls which together provide benefits. These controls are more about avoiding pest problems rather than killing the pest, and the first three are about getting the plants growing quickly so that they spend as little time as possible in the vulnerable cotyledon stage. So our first control, planting early after the autumn break. That is, planting before the temperature drops and slows the rate of growth. Good quality seed and soil preparation. So good quality seed and good contact between the seed and the soil promotes rapid germination and growth. These are points that come up regularly in discussions with farmers and agronomists. Our next control, stubble retention, which provides habitat for pests and beneficials. So the effect of retaining stubble will actually be seen over several years. It's not seen as an immediate benefit in terms of predatory species. And finally, weed management. Controlling broadleaf weeds in winter, the year before the canola crop, will reduce the number of pests such as red-legged earth mite. Controlling flowering brassica weeds will reduce the nectar sources for diamondback moth and cabbage white butterfly, and so reduce the number of eggs that they lay. Here's our final example, cultural controls in cereals. The control methods that we just outlined for canola are also mostly apply here. So control methods include planting late in autumn rather than early. So this is done to avoid the main aphid flight periods, so the crop's not exposed to high aphid pressure and possible barley yellow dwarf virus pressure. This may eliminate the need for insecticides. Another method uses barley yellow dwarf virus resistant varieties. Soil preparation, of course, is important, using good quality seed and weed management. The photos shown here show barley yellow dwarf virus in wheat, which shows the classic infection early on of the flag leaf. And the second photo shows the infection in a crop of dagger wheat. Now let's move on to chemical controls. Chemical control in IPM is used as a support tool only. It is not the primary control tool. The registration of pesticides rests with the states in Australia, although there is a national registration authority. So a farmer in Victoria, for example, could use an insecticide called Success or Entrust to kill caterpillars in a crop of canola or wheat, but it's illegal for a farmer in New South Wales to do the same unless a permit is issued. In Victoria, farmers can use the pesticides for other situations, but at his own liability. That is, if it doesn't work, he can't sue the chemical company. Entrust is used by organic farmers because it has a minimal effect on beneficials, non-targets, humans, birds and fish. But Entrust is not registered for use on broadacre in New South Wales. New South Wales farmers can use a range of synthetic pyrethroids or organophosphates that are non-selective. And because they're non-selective, it means that they may be toxic to a wide range of animals, fish, birds and mammals. So the argument on pesticide here is not logical, but it's bound by law. If we consider the conventional pesticide approach, most information is readily available from the Australian Pesticides and Veterinary Medicines Authority, APVMA. But the problem is there's no demand from farmers to know information on the effects of these chemicals on beneficials. So it's difficult to source this information for the IPM approach. So generally it's recommended in IPM to avoid broad spectrum chemicals which leave the highest amount of residue, and this includes synthetic pyrethroids and the organophosphates. There is information available online on the side effects of pesticides on beneficials in horticulture, and this information can be extrapolated to broadacre farming. The impact of chemical pesticides is not equal on all species or on groups of beneficials. The effects might be short-lived or they might be residual. So with acute poisoning as a result of pesticide use, individuals either live or die in a short time period. But when the effect is chronic sublethal, beneficials can be affected by the chemical pesticides for days, weeks, or even months. Some individuals might survive, but their reproductive ability or fecundity is affected. So this might affect the number of eggs or the viability of their eggs. And this, of course, disrupts the population of beneficials. So in other words, individuals that are exposed to chemicals might survive the dose, but the population is affected. Another problem is that beneficials may eat treated pest insects. These are pest insects that survive the pesticide. For example, they might be in a sheltered location. So the beneficials then start to accumulate the poison. Other pesticides actually kill all the individuals but have no residual effects. And this means that re-immigration of beneficials is possible. This is much better than a long period of preventing beneficials from re-establishing. The same chemical can be applied in different ways. So it could be applied as a broad spectrum crop spray versus a seed dressing. The broad spectrum spray would kill a range of beneficials, but when used as a seed dressing or bait, the effects would be very much reduced, and so this could be part of an IPM strategy. Using the chemical pesticide as a border spray to prevent pest invasion would kill pests and beneficials alike, but most of the beneficials in the paddock would survive. So even broad spectrum sprays can potentially have a place if used in certain ways. It's difficult to actually provide a list of safe and unsafe chemicals. We need to think about every pesticide application, what we are targeting, how we are targeting them, and the consequence of that application. So how do we respond to claims of safe to beneficials and safe in IPM programs? How do you decide if a product is safe for use in agriculture? The reality is that chemical companies are much more skilled in testing the effects of chemicals on pests rather than on beneficials. To really be considered safe, the chemical should not kill as spray droplets, not kill after exposure to dried residues, and not reduce fertility or egg hatch. That is, egg hatch after exposure to either the spray itself or to eating treated prey. And these tests are not conducted as a routine measure, but hopefully this will change in the future. So information about sublethal effects is severely lacking in Australia. For example, nicotine and thalidomide would be passed as safe to humans if only 24-hour acute testing was conducted. So we should actually seek information from other sources, such as private companies and universities. How do you decide? There are three broad categories of pesticides in agriculture. The first group includes insecticides, miticides, and molluscicides. Group number 2, the fungicides, and 3, herbicides. The first group here is of the most concern, as they're designed to kill certain groups of animals, so they're more likely to impact on beneficial insects than group 2 and 3. We can't assume that because a pesticide is allowed for organic agriculture, that it is also suitable for IPM. For example, pyrethrum and rotenone are derived from natural sources, so they're permitted in organic agriculture. But the problem is they both kill a wide range of beneficials, and so they're not desired in IPM strategies. IPM is based on biological and cultural controls with support from chemicals of any origin. So this is true for both organic and conventional agriculture. So the only difference between organic and conventional IPM is the list of support chemicals available. Apart from that, there's very little difference between the two. Suitable selective products are not always available, as there's no demand from chemical companies. What about the broad spectrum lannate, which is permitted on crops with only a 1-day withholding period? But the problem is it kills for 12 weeks. So the registration of products and their withholding periods are not a reliable guide as to their usefulness in IPM strategies. First choice should be to always look for pesticides that have a short residual activity. So even if all beneficials are killed, at least they won't impact on the newly arrived beneficials. Monitoring, what to look for. First signs of pest problems are symptoms of damage rather than observations of pest numbers. And it's important to recognise that several different causes may lead to almost identical damage. So it's important to look at each site and identify just what is causing the damage. There could be more than one cause. For example, a canola crop could have slugs, earwigs, birds and beetles causing damage at the same time. Which of these are the primary target? Usually there's one or two. Other factors should be considered. Herbicide burn, seed dressings, hail and waterlogging can all cause responses similar to pest damage. The role of monitoring is to provide the farmer with information about pests and beneficials in a paddock, so they can make an informed decision about what, if any, actions are necessary. Regular monitoring constantly updates that information, so decision making can be precise and timely, but it's dependent on correct identification of the cause. So it's crucial that the person monitoring is able to make the correct ID. Important point here, presence of a pest does not mean there is a problem. If you're just starting to implement IPM, start with just a few paddocks. Select 1 or 2 paddocks as trials for IPM. Choose paddocks that are not high risk at first and begin to apply a set of compatible control measures for all the pests. This enables confidence and experience with pests, pest pressure, beneficials and other invertebrates. Consult an experienced IPM advisor. How do you monitor your chosen paddocks? The range of monitoring methods depends on the type of invertebrates. So knowledge of life cycle and habits of each species allows you to use trapping or sampling methods to target each species. Knowledge of history of pest damage in a paddock helps decide how many monitoring points are necessary and where to actually place them. It's impossible to monitor a whole paddock, so look in wetter areas for slugs or borders for mites, or in the absence of this type of information, choose a convenient location and move it around over several weeks. Monitoring methods include the use of yellow sticky traps to monitor numbers of small flying insects, including aphids and brown lacewings. Direct searching is very useful for earwigs, slugs, carabid beetles, cutworms and weevils in soil, aphid colonies, moth eggs and leaf damage. The disadvantage with sticky traps is that they attract a huge range of flying insects, including flies, and so looking for a particular species is time consuming and the traps tend to get covered with dust and dirt. And note that aphids caught may not be the type that will colonise a cereal crop. Direct searching depends on the person's eyesight, skill, patience and experience, and also the time of day and weather. However, if the same person conducts the searches, then the results are more consistent. Monitoring methods also include the use of pheromone traps. These are used for monitoring the activity of some species of moths, and pheromones, of course, are chemicals produced by the female to attract the male moths. Pheromone traps for Etiella moths of lentils specifically attracts Etiella, and they last for about 1 month. Just the bait is supplied, the trap or holder needs to be constructed about 250 millimetres above the canopy and 2 to 3 traps placed per crop. Pictured here is the Etiella moth. Also pictured here is a light trap, but this is probably not as useful as it attracts all types of flying night insects. Traps should be emptied once a week and the number of moths in each trap recorded. Sweep nets are commonly used to sample the upper part of some crops. This is useful in order to estimate the number of pests or the number of beneficials. So sweep net is a standard tool of crop monitoring. Useful for pests including weevil and caterpillar. Sweep net sampling is standardised by sweeping a set distance, for example 25 metres, or taking a set number of swings, for example 25. And pictured here is one of the current methods used for sweep net sampling, but note there are many different methods used. Pitfall traps are useful to collect invertebrates active on the soil surface. Alternatively, shelter traps can be used, for example tiles or sacks. Pitfall traps can be filled or partially filled with antifreeze, which acts as a preservative, with a lid to stop it filling with rainwater. It's placed with its rim just above the soil surface to prevent rainwater from filling the cup, and a funnel can be used to prevent escapes, as pictured here. Monitoring methods ideally should begin a year or two ahead of a vulnerable crop or pasture being planted. Monitoring within the crop should take place weekly during critical periods of pest activity. So, for example, after the autumn break, during spring flights of aphids, and during spring and summer flights of moths. Keep records. Keep a paddock diary where you list dates, seasons, presence of pests and beneficials, record any action taken and the results of those actions. So these records can be used in future years regarding the timing of flights, which may be seasonal and can then be predicted, as can the movement of beneficials and how populations are developing. Compare last year's information to this year to assess whether pest pressure is higher or lower. Outcomes can then be assessed. What are the outcomes from monitoring? There are three possible outcomes. 1, there are no pests and so no economic damage. 2, there are many pests and likely serious economic damage. 3, there are some pests, some beneficials, damage is possible. It's easy to deal with outcome 1 and 2, and an appropriate action is easy to decide on. So outcome 1 is surprisingly common and may reduce pesticide use just by monitoring and avoiding sprays just in case. Outcome 2, action must be taken that is effective with the least impact on beneficials. The third outcome is more difficult to assess. If the outcome of monitoring is outcome number 3, try to answer the following. Pests are present, but is there immediate damage? Is there immediate danger of economic damage? What is the level of damage to plants, and will they grow out of this risk stage soon or not? Is the damage going to cause economic loss? So when should you use a pesticide? Try to consider these questions. What is the pest present? Are there beneficial species present, and what are they? What other pests are present? What other beneficials are present for those pests? Are there sufficient pests to cause economic loss? And are the beneficials likely to control the pest in the short term or long term? If it's been identified that the pest requires control, or if the farmer is facing economic loss, consider the question, are there selective pesticides available to spray? If yes, spray. If no, or yes but too expensive, consider this question. Is a seed dressing or bait or border treatment possible? If so, this would be the next ideal control method. Also think about the final two questions here. What beneficials will be killed by the non-selective pesticide, and what pests may occur as a result? Finally, what is the cost to control these pests in the near future or next crop? Choosing the pesticide with the least residual activity allows the beneficials to reinvade more quickly. So try to get information on what residual toxicities the pesticides have. Finally, did the pesticide perform as hoped against the pest, and what was the effect on the beneficials? So important decisions for farmers to make. Do we continue to use conventional pesticides? If no, assess an IPM approach. How do we implement an IPM approach, and how do we get the information required?